I think we're going to get started here. Thank you, everybody, for joining the last scientific seminar of 2021. I'm still in the COVID time warp. This is our last seminar of this year, and I hope you'll join us again when we start back up in the spring, starting on February 3rd. Today we have Dr. Cynthia Downs with us from SUNY ESF. She is actually here in person just down the hall, but we are joining virtually so that we can have a wider audience. We're still waiting for our auditorium to be back up to speed so we can do it all, do virtual and in person at the same time. Dr. Downs is faculty in the Department of Environmental Biology at SUNY ESF, and I believe she started there maybe just a few months before lockdown happened. So it's been a bit of a tumultuous start, but I actually met her when she was an assistant professor at Hamilton College, which is not too far away from us in Clinton. And that's where I became familiar with her research program and I realized that she was really also a very gifted mentor. She had a small army of undergrad mentees who, were clearly just inspired by her work and her teaching. And I actually got to mentor one of her former students as a summer undergraduate research student here in my group. He was a National Science Foundation research experienced and undergraduate student, and he credits you, Dr. Downs, as being one of the best professors he ever had. So I think that's a testament to your giftedness as a mentor. Dr. Downs did a postdoc at Ben Gurion University of the Negev in Israel. And before that, she completed her PhD at the University of Nevada in Reno, which she earned following her BS in biology from SUNY ESF. So she's now returned back to SUNY ESF, her alma mater, to continue inspiring students there. Dr. Downs is an ecological physiologist, and in her work, she combines perspectives on animal physiology across species, and applies this comparative approach to try to understand the ecology and evolution of immunity and what this means for the ecology and evolution of infectious diseases. And what I really appreciate about her work is that she continues to publish work that really explores some pretty fundamental questions about what differentiates the way that animals contend with pathogens. And it's a question that's becoming increasingly important. Why did they evolve the strategies that they did, and what could this mean for their ecology? So without further ado, thank you so much for joining us. We're really looking forward to hearing more about your work.
Thank you. Just give me a second to get the presentation up. Okay, I believe you can now see.
Yeah, that's great.
Great. Thank you for that kind introduction. I'm really excited that my first in-person talk after the beginning of the COVID is here at the Carey Institute. I think the work that comes out of this institute is amazing, and it's really inspired some of my thinking about how immune defenses are shaped. So today I'm going to be talking about how body size shapes immune defenses. And I think a good place to start is thinking about host competence. And I probably don't have to tell this audience that host competence is the capacity for an individual to infect another individual with a parasite. And I should start by mentioning that with my training as an ecologist, I tend to use the word parasite broadly. So I use it to encompass viruses and bacteria that are pathogenic, but also to encompass the more traditional ectoparasite, macroendoparasite use of the word. And host competence, as I said, is the ability for an individual to infect another individual with a parasite. And this can be broken down into four multifaceted components. So first, an individual would need to be exposed to a parasite in the environment. They need to be susceptible to that individual or that parasite. That they need to be suitable for replication and reproduction of that parasite, and then they need to transmit that parasite to another individual in order to be competent. And as a person who's interested in immune defenses, I really focus in on this suitability component because as you know, the immune system evolved to be a defense against mutated self cells, but also parasites and pathogens. And so it's that frontline defense. It's the kind of physiological barrier between a host and an infectious pathogen. And I think because I'm interested in what's driving variation among species in this suitability and the immune defenses that underpin this suitability, I'm going to make a case for body size being an important component to think about in a comparative framework. I think that we can, you're right to start with the question of why do we expect immune defenses to change with body mass? And I can actually point to work from here at the Carey Institute as evidence of why we should start thinking about body size. So we see diversity in host competence that changes with body size. So Rick Ostfeld's work in 2014 from his team looked at reservoir competence for Borrelia burgdorferi, the agent, bacterial agent that is one of the bacterial agents that's responsible for Lyme disease, and found a positive relationship between pace of life and reservoir competence, such that fast-paced species were more competent. And fast-paced species have a low annual survival rate and a high annual reproductive rate, and they also tend to be really small. So this is sort of indirect evidence that body size might play a role in competence and underpinning suitability and immune defenses. And then Barbara's work in Journal of Animal Ecology from 2015 performed a modeling exercise to look at the base of reproductive value of macroparasites and microparasites, and used how ecological characteristics of species change with body mass to model that microparasites increase positively in ungulates and primates, and that macroparasites show a negative relationship with host body size in carnivores, primates, and ungulates. We also have evidence that body size mediates organismal traits that are associated with immune defenses. So I have examples of two of the many here, but one of them is a physiological trait, basal metabolic rate, and these are basal metabolic rate data from mammals and birds, and it shows a disproportionate relationship with body mass, such that 1 gram of elephant tissue has a lower metabolic rate than 1 gram of mouse tissue. And this is whole organism metabolic rate depicted here. And the metabolic theory of ecology would argue that metabolic rates, basal metabolic rates, set the pace of biological processes, including cell replication and protein synthesis, which are critical for, that are important processes when in immune defenses. And then here are data on social group size in relationship to body mass. And again, there's a positive relationship. And this should affect, social group should affect this ecologically important trait should affect how the probability of an individual encountering another conspecific that's infected with a parasite, and therefore transmission probability, which again should shape the ecology and the evolutionary framework that is shaping immune defense variation among individuals. And then we also know that host body size affects parasite community in sort of a predictable manner, and it's actually predictable from island biogeography theory if we think of hosts as islands. And research, published research shows that large hosts have higher parasite abundance, biomass, diversity, size diversity, and prevalence. And here on the left, I have a graph showing parasite biomass in relationship to host mass. And you can see that larger hosts have a greater biomass of parasites on them and in them. And then on the right, I have a graph showing parasite body size. And here this is evidence that large hosts have disproportionately larger bodied parasites residing on them. So this sets up an argument to suggest that scaling, or how traits change with body size, might provide an overall organizational and comparative framework for thinking about the immune defenses. And this should then inform our ability to build disease ecology models and ask questions about spread of emergent diseases in new communities. And it should also help us think about how to translate biomedical research performed on small model organisms to human health. Now, for those of you who don't sit around thinking about regression lines and how traits relate to body size, let me start with a quick primer in how we perform scaling models. So traditionally, they're modeled as power laws such that Y, the trait of interest, is proportional to body mass raised to little b, the scaling exponent. And if a trait changes as we'd expect, based on the geometry or underpinning physics, is said to be isometric. So here on the y-axis, my trait of interest is an immune defense that's a concentration, and the isometric prediction for little b, which describes the shape of the line, would be 0, because we would expect that, for example, a gram of mouse tissue might require the same number of cells as a gram of elephant tissue. But we don't often see isometric scaling. Instead, we see allometric scaling, which is disproportionate scaling or disproportionate changes in the trait of interest, such that, for example, in hypermetric scaling, a large organism, in this case, my cartoon elephant, would have a disproportionately higher concentration relative to a mouse or a small organism, and B would be greater than 0. And if B is less than 0, we see this hypometric pattern in blue, such that an elephant has a disproportionately smaller concentration than we would expect based on isometry. And humans are really not great at thinking about nonlinear relationships, and especially in these power laws. I think the early predictions out of the COVID epidemic are a great example of that. You know, the news outlets were saying it's not growing that fast in the US, it's not spreading that fast in the US. And then a week later, we were going into lockdown because we were in the nonlinear part of an exponential curve. And so what people who have been studying allometries and scaling have done is that we convert things on a log-log. We plot on a log-log axis such that the equation becomes the log of the trait of interest is proportional to what is now termed the scaling coefficient times log of the body mass. And all this does is it means that the scaling coefficient, or little b, now depicts, or tells us about the slope of the linear line on the log-log scale. And the same patterns hold. Hypermetric is a b that is greater than 0. Isometric is a b equal to 0 for a concentration, and hypometric is a b less than 0. So today I'm going to talk about some work that's been coming out of my lab the last couple of years. I'm going to talk about 2 types of scaling of immune defenses. First, I'm going to talk about scaling of cellular morphology, or concentrations of immune cells. And then I'm going to talk about scaling of a functional serum-based immune defense. And then I'm going to tie this back to host-parasite interactions at the end. The hypotheses that underpin all of my work, there's 4 hypotheses about scaling of immune defenses that underpin my work. The first 2 hypotheses, the protect-on hypothesis and the immune complexity hypothesis, both lead to a prediction of isometric scaling, or a B that is equal to 0. And through different lines of logic, they argue that each gram of tissue needs the same concentration for protection. The rate of metabolism hypothesis is an extension of the metabolic theory of ecology, and it argues that if metabolic rate is the pacemaker for biological processes, we should see immune defenses scale in a manner similar to mass-specific basal metabolic rate, which has a slope of negative 0.25, and we would predict hypometric scaling for immune defenses. The 4th hypothesis was developed by my research group, and it's the safety factor hypothesis. It's an extension of performance safety relationships from biophysics and physiology, and it argues that larger species should have evolved disproportionately stronger damage mitigating mechanisms than smaller ones, because of the hypermetric scaling of discrepancies in replication rates, because of the pacemaker ideas of metabolic rate, and also because larger organisms are disproportionately exposed to parasites because of their how home range size scales, and how the locomotion of these organisms scale, among other things. This leads to a prediction of hypermetric scaling of immune defenses, or a B that is greater than 0. So first I'm going to talk about scaling of mammalian cell concentrations. And this work was conducted with Marty Martin, Ray Ball, Ned Docterman, and Kirk Klasing. And we focused in on neutrophils. So neutrophils in mammals are often the first site to a cell to a site of infection. When a pathogen is detected, it sends out chemical signals that recruits neutrophils from the circulatory system to the site of infection along a chemical gradient. They follow a chemical gradient. And once at the site of infection, neutrophils can undergo 3 antipathogenic activities. The first is phagocytosis. They engulf the pathogen, and that phagocyte or that phagosome merges with vesicles full of antimicrobial molecules and also reactive oxygen species that kill the pathogen. The second is degranulation, and degranulation, these cells are full of little granules full of toxic chemicals, and they release those toxic chemicals into the extracellular fluid where it can then attack and kill the pathogen. And the third, which I think is just the coolest one, is that they can actually release their chromogen, their DNA in the form of chromogen, and create this net that captures the pathogen, and that net is also full of antimicrobial molecules that degrade the pathogen. And that process is called netosis. These are also really important. These cells are really important links between the innate and early immune response and the adaptive immune response, because they help regulate inflammation, which is a later innate immune response, and also the activity of B and T cells that are involved in the adaptive immune responses. So these cells are really critical to the outcome of a pathogenic infection, and there are these early acting cells that are important. So we collected data using from the ZIMS database. This is a database of adult healthy organisms that are housed in zoos, zoo facilities, and it's a reference database used by vets to determine if the values they're getting from their animals are within the normal range. And we have data from 259 mammalian species, and we found that neutrophils scale hypermetrically in a manner that is consistent with the safety factor hypothesis. So the slope of this line, which was obtained from a phylogenetically informed MIC generalized linear mixed model, general linear mixed model, is 0.011. And that may not seem like a big deal because it's really shallow when we think about it, right? A slope of 0.11 isn't that big of a deal. But if we take these concentrations and we do some calculations to calculate how the concentrations or the number of cells per liter scales up by multiplying it by the liters of blood an organism has, we can get to the total number of circulating neutrophils. And what we found is that for the difference between a mouse and an elephant, there's a 250,000-fold difference in body size, but the change in neutrophils is a 13.3 million-fold difference in neutrophils. So I've come to think of elephants as these sort of bags of neutrophils. And this is just a disproportionately high number of neutrophils, and leading to the question of why, right? And so this is actually the data that we use to develop the safety factor hypothesis, and it's consistent with it. So you might ask, well, that's mammals. What's going on with other organisms? And so we, Emily Roos, Dr. Emily Roos, a postdoc in my lab, who's now at University of Chicago as a postdoc, did a similar analysis looking at heterophils in birds. Heterophils are functionally equivalent to neutrophils, and using data from 116 species, again from the zoo reference database, she found that these scaled heterophils in birds scaled even more steeply. So the slope is 0.19, and this is again is hypermetric and consistent with the safety factor hypothesis. It is, though, steeper than mammals. So again, let's do some back of the envelope calculations and think about how this relates to total number of granulocytes, either heterophils or neutrophils in birds and mammals respectively. Well, what we can do is we can look at two similar sized small mammals and birds, and look at two similar sized large mammal and bird, or mammal and bird. And for an increase that is a 1,200-fold difference in mass in both mammals and birds, what we see is that the birds increase their granulocytes by 4,500-fold, and the mammals increase their neutrophils by only 2,600 fold. And so what this means is that our equal-sized hyena and emu have, the emu has twice as many circulating granulocytes relative to the hyena, even though they're the same body mass, and that the small bird and mammal have the same number of circulating neutrophils. So this opens up the question of what's going on in birds. Why do they need disproportionately even more protection when they're large relative to when they're small and relative to their large mammalian counterparts? And so we think that there might be a couple things going on. We propose that it might be differences in their immune system. So birds don't have lymph nodes and mammals do, and maybe the absence of lymph nodes mean that they need more surveillance cells because they don't have sort of these localized hubs that their cells can leave from, and that can act as integrating centers for the immune system. The heterophils from mammals are missing one of the types of granulocytes that we find in mammals, and the antimicrobial molecule found in that granulocyte is actually one of the most potent antimicrobial molecules in mammals. So maybe they need more cells because they're less potent. And then, of course, an argument that always comes up when you think about differences between birds and mammals is that maybe it's related to evolution of flight. So what Emily did next is she went and looked at the role of flight, and these data are proportions rather than concentrations because we couldn't get concentration data from bats, but she compared the relationship, the scaling relationship of proportions of these granulocytes in birds, bats, and non-volant mammals. And what she found is that we don't see, we see isometric scaling for sorry, this graph is labeled incorrectly. This should be neutrophils. We see isometric scaling for neutrophils in bats, and we still see the hypermetric scaling for proportions of heterophils in birds and also a proportion of heterophils for non-volant mammals. And what this suggests is that it's not just flight by itself that's driving these differences. Rather, it's something unique about birds. The other question that comes up regularly when I present these data are what is the role of captivity? So here I have a picture of on the left of an elephant in a zoo, and I have a picture on the right of an elephant in, I think this is Uganda. And you can see that the environments that they're inhabiting are fairly different. And we know that the developmental environment of organisms is really important in shaping their immune responses. So in general, birds that are in the, or animals in the wild have a more robust and diverse immune response than animals that are raised in captivity. And you can imagine that the pathogen exposure of an elephant in a zoo versus an elephant roaming free in Africa are going to be are going to be different, right? They're in a different parasitic envelope, so to speak. So what we did is we collected data from birds, again, data from the literature and from the ZIMS database on proportions of cells of animals in captivity and animals raised in the wild, and looked at whether or not the slopes differed. Since what we've been talking about throughout this talk are slopes. So again, these data are proportions because that's what's available in the literature for wild animals. And for lymphocytes, which include, is a classification of cells that include B cells and T cells and natural killer cells, we tend to think of it more as the adaptive branch of, cellular adaptive branch of the immune system. We see that the scaling, the slope and the regression line for captive and wild animals doesn't differ. There's no statistical evidence that it's different. And if we look at heterophils, we see that the captive animals actually have a higher intercept than those of the wild animals. But again, the slope, the scaling coefficient for these two lines don't differ. and they're still both hypermetric. So this suggests that at least for some immune defenses, that wild and captive animals don't differ in their slopes, and that we can draw conclusions cautiously from our information about captive animals. And just to kind of step back, the reason we're using data from captive animals is that it's this nice, robust data set. And in my next set of information, I'm going to move towards an experimental or a lab-based system where we looked at a functional immune response. And working with captive animals allowed us to kind of draw samples from animals that are in more standardized housing conditions, and so hopefully reduce the between individual variation within a species. The next thing we did is we're now moving on to that second bullet point of scaling of immune defenses. And we looked at functional antibacterial immune defenses. This work is from an NSF grant that is co-PI'd by Marty Martin and Ray Yang down at University of South Florida. And then the work was also conducted with the help from Kurt Klasing, Sam Oki, sorry, I'm over top of my own photos at this point, Laura Schonley and Ray Ball, who, and Laura was a postdoc in the lab at the time. Give me 1 second. This work is really focused in on asking about the functionality of the complement cascade. And so to take a very complex cascade, biochemical cascade, and to overly simplify it, I'm going to summarize it this way. So there's an invading cell, and the invading cell is coded with these pathogen-associated molecular patterns. And the immune system of vertebrates has evolved to recognize these pathogen-associated molecular patterns, and it produces molecules that bind with those pathogen-associated molecular patterns. And these molecules act as opsonizing agents and basically mark the cell as foreign. And they include things like antibodies, but can also include other molecular markers, and they activate the complement cascade. And the complement cascade actually has 3 outcomes. But for the purposes of this, I'm asking about the functional outcome of the building of a membrane attack complex. And the membrane attack complex through this complement cascade is built through this complement cascade, and it basically creates a hole in the invading cell and allows water to rush in, and it lyses the cell. And so we're looking at lysing, the end result of this pathway. So we developed this antibacterial capacity assay by adapting it from the work done by French and Newman Lee. And it basically is a measure. We take serum from an individual, and an advantage of this is we can actually use frozen bank samples from zoos. We take serum, we mix it with a growth media, and then a concentration of a microbe. We used 3 microbes for this assay. We used E. coli, a micrococcus, and a salmonella. I'm going to focus in on the E. coli today, but it is representative of the results that we got for all of the testing the antibacterial capacity against the other 2 microbes also. And what is functionally happening is the complement cascade is both lysing the cell, but it's also inhibiting the microbes from being able to grow and replicate. And so what we're seeing is the end product of those 2 processes. And so that's what we're calling antibacterial activity. And what you would normally do if you were doing this on a single species is you would run a dilution curve like the 12-point dilution curve seen here, and you would try to find a concentration where about 50%, there was a 50% antibacterial activity relative to a control grown without serum that worked for a pooled sample of that species. When you try to do this cross-comparative work, it becomes a little more complicated. So what I have depicted here to just pick the problem is a curve from an elephant and a Malaysian tapir. And what you can see is that the elephant has 100% antibacterial activity until a very, very low dilution of serum. And then it drops down. There's a red dot here behind everything else to 0% antibacterial activity. And that's a wildly different curve from the Malaysian tapir, which starts at about 20% or 25% killing, and then has a gradual shift towards 0% antibacterial activity. So what we did is we ran these 12-point curves for every sample that we got. And then we could run 2 curves per plate. We ran them because we ran them in 12 points and in triplicate. And that 12-point curve is what we're calling antibacterial capacity. The other thing we did, though, is we ran a cow curve of 4 dilutions that spanned from 100% to 0% antibacterial activity on every plate as a control. Once we have those 12-point curves, we can fit a nonlinear curve to it and extract the curve parameters, which we can use in a multivariate model to ask about how the shapes of these curves are changing with body size. And so we can look at the maximum antibacterial activity, which is the top asymptote. We can look at the proportion killed or inhibited at the inflection point as a measure of antibacterial activity at a specific amount of microbe. And we can look at the slope of at the inflection point, the instantaneous slope at the inflection point, to look at the rate of switch between maximum and minimum. So going back to these 2 curves previously, the elephant has a very steep slope at that point, and the Malaysian tapir would have a much more gradual slope at that point. We can then take these curves and we can make predictions about how they should change. And here I'm going to use a hypothetical elephant and mouse as my examples in relationship to these hypotheses. And so the protecton and the complexity hypothesis, which predicts isometric scaling, would predict these lines lie directly on top of each other. These curves lie directly on top of each other for a mouse and an elephant. The rate of metabolism hypothesis, which predicts hypometric scaling, would predict that a large animal relative to a small one, so the elephant relative to the mouse, would have a lower maximal antibacterial activity. It would have a lower specific antibacterial capacity at a specific dilution, and it would have a more gradual change from maximum to minimum. And the safety factor hypothesis, which predicts hypermetric scaling, would predict the exact opposite for our large and small organisms. To the right of the slide, I have a picture or pictures of the species we included in this study. We worked with 22 zoos and research groups to get to collect our samples from 1,256 unique individuals representing 199 species, 164 genera, and 60 families of mammals. We have a mean of 4 individuals per species, and for an analysis that has not been done yet, looking at inter versus intra-specific variation, we have data for 16 species across this body size range where there are at least 15 individuals. We restricted our analysis to samples collected between 2005 and 2019. The zoos were extremely generous with sharing samples with us, and I cannot thank them enough. Our smallest species was a 16-gram old field mouse, and our largest was a 3.6 million gram African elephant. And this is a 223-fold difference in body size, and it also spans the range of body sizes of extant mammals. As I said, we used a MCMIC GLMM model to a multivariate analysis to account for the shapes of the curves and how they changed with body size. We actually did this analysis 3 different ways. I'm only going to present these data, but we did do a phylogenetically informed version of this analysis also. And phylogeny actually explains a very small amount of the variation. And so I'm going to focus on the non-phylogenetically informed version of the analysis. And to cut to the chase, we see isometric scaling of this antibacterial capacity. So all of our slopes are have credibility intervals that overlap 0, suggesting isometric scaling. And so our interpretations are that there's equal antibacterial activity with the same volume of serum. We see equal antibacterial activity at raw serum, or that maximum antibacterial activity. And we see an equal transition between high and low antibacterial activity. So the slopes at the instantaneous slopes at the inflection point are the same. Now, this is not to say that all mammals have the curves that look exactly the same. We saw really big differences in curves, but those differences were not explained by body mass. And just to depict some of these data in a more traditional fashion, what we have here are species means of the slopes for all of our species. And you can see that it is a pretty flat line, and it's isometric scaling. So these data from the antibacterial capacity support the protecton or the complexity hypothesis. And I say this with a couple caveats. First, the protecton and the complexity hypothesis predict isometry. And isometry is the null prediction. And so it's hard to find support for a hypothesis and interpret support for a hypothesis when it's the null expectation. But these are consistent with, these data are consistent with the protecton and complexity hypothesis. The other thing I want to point out, though, is that it is larger organisms do have a higher antibacterial capacity than is expected by metabolic rate alone, or the pace. If metabolic rate acts as the pacemaker for cells. And so what we did is we measured complement, and complement is produced, proteins are produced mostly by liver cells, and liver mass scales hypometrically. And so we would expect, what this suggests to us is that fewer in a larger organism, a disproportionately small number of liver cells are producing the same amount of functional amount of the complement response as in a small organism. And so this lends the question, an open question of how are large organisms producing more complement per gram of liver tissue? We're in the process of, we've written a grant recently to try to explore some of these questions at get down into the mechanism of driving some of these differences. The other thing I think that is interesting about this is that the antibacterial capacity is scales similarly to a measure of the ability to recover from an immune challenge and an inflammatory response, which is a measurement of antioxidant activity. So with Dr. Jimenez at Colgate University and a whole army of undergraduates, we measured antioxidant activity, among other things, in 28 species of birds and 48 species of mammals. And again, using phylogenetically informed statistics, we found isometry, right, for antioxidant activity. And so I think it's kind of interesting that we have some immune responses that are scaling hypermetrically, and we have some that are scaling isometrically. And I think it's particularly interesting for something like antioxidant activity, because the neutrophils, one of their things that they do in their degranulation is that they actually release free radicals and they are involved in this oxidative burst. And that can lead to damage of host tissue as an artifact of trying to kill a pathogen. And the ability to mitigate that damage through antioxidant activity seems to scale differently than the propensity to cause that damage based on the number of cells of neutrophils that are circulating. And so I wonder if there's maybe something like a passive tolerance going on here, where large organisms, because they have so many more cells, say in a hypothetical that the release of antioxidants from one neutrophil kills 3 cells, and an elephant kills more, right? It would kill more cells because there's more neutrophils, but maybe it's okay that that damage isn't repaired as quickly because it's a smaller proportion of cells in the elephant relative to the proportion of cells in a mouse that are damaged at that response. And so this has implications for host-parasite interactions. We know that larger hosts have different parasite communities, and those parasite communities may be partially different because large and small organisms have different immune strategies. So as I said, these large organisms have disproportionately high neutrophils and heterophils, but they have proportional antibacterial and antioxidant activity. And I'm now in the process of trying to think about what this means for sort of a overall strategy of immune defense. We know that they're using different strategies, but we don't know if those strategies have different functional outcomes when we scale back or when we look at the actual immune defense as a whole. The other thing is I think that this work has implications for understanding the spread of multi-host pathogens in a community. So previously we published a paper proposing a scaling framework for thinking about host competence and predicting host competence, which basically involves taking these 4 steps of host that underlie host competence and the processes that underpin them, and thinking about the scaling relationships of those processes. And if you take those and do the math, you can end up with a prediction for how host competence should change with body size, utilizing over 100 years of data, published data on how organismal traits change with body size. And it leads to some interesting predictions, but actually one of the biggest gaps is in the susceptibility component, because we seem to be missing a lot of information from a comparative about immune defenses from a comparative perspective. The other thing I think this has implications for, as I mentioned in the beginning, is kind of recentering our perspective about human health towards adaptive responses. So we tend to think of, we see things like inflammation, and we see high neutrophil numbers in wild organisms. And because we have immune responses have been, our understanding of immune responses have been developed in sort of a human disease framework, we tend to think of those as being maladaptive, but they should have been shaped by evolution, just like all other organismal traits. And so they should actually be adaptive for the parasitic environment that that organism is living in and its evolutionary history of its host parasite interactions. And so I think by taking this comparative approach, we can start to think about how animals are have evolved, and their immune systems have evolved in providing sort of an organizational framework for thinking about immune defenses in a comparative way. And I think this has implications for understanding how studies on mice, a small, fast-paced organism, scale up to understanding human responses and interactions between humans and their parasites. So I've thanked people throughout, but I really want to highlight the undergraduates who helped collect the data for that functional-based immune response. They're pictured and named here. And then I want to provide a special thanks to Nikki Keith, who was a postmaster's student who came and worked in my lab and helped develop the R code to process data from over 1,200 individuals. Without her, I would still be trying to figure out how to bind files together. And then I also want to thank Sam Oki, who ran the lab, Marty's lab down at USF, and really helped make sure that the samples processed there were processed. And thank you to you for taking the time to listen today. I think we have some time for some questions. Thank you.
Thank you so much, Cynthia. I'm going to change my view because I can't see you. Okay. We have a question here from me. I have a bunch of questions. I thought that was such a fascinating talk, and I actually had never heard of netosis, the ejection of the DNA to capture a pathogen. So I'm definitely going to ask you about that later. Yeah. We have some raised hands actually, so I'm going to see if I can manage to do this somehow. Olivia, I see your hand raised. I'm going to just unmute your mic so you can talk, Olivia, or not. She disappeared. Okay. Sorry, I may have messed that up, but I'm gonna field the question here from one of our attendees that she asked in the Q&A. She asks, have you examined differences in migratory versus non-migratory bird or mammal species in regard to the heterophil or granulocyte concentrations? And she follows that up with the question that is related, which is, what about migratory and non-migratory populations of the same species? So things like Florida whooping crane populations that are resident versus migratory.
Yeah, that's a great question. Presumably, that's going to shape exposure to parasites, right? Migration can act as a filter that kind of eliminates, for lack of a better term, sick individuals, but it also means that individuals are being exposed to whole different communities of pathogens as they're moving through their migratory route and in their summer and wintering habitats. It's a great question. The short answer is no. The long answer is it's one of the things that Emily is really interested in doing, and she's been compiling the data set to do. And I'm not sure, as I said, she's left to move to gone to a new postdoc, and I'm not sure the status of that work at this point, but it is something that is on our radar. I think asking about the ecological characteristics that are shaping some of these patterns is one of the next questions.
Yeah, I think I overlapped with Emily at the University of Georgia, and she might have been working on the migratory butterfly system.
Yes,
yes. And that's one of the interesting hypotheses there is that, you know, the species that are resident just they are paying a price for being residents in terms of being more susceptible to death from pathogen, or they can't make the migratory journey because they aren't able to escape the pressures of, or I guess handle the pressures of the migratory journey. So yeah, it'd be really interesting to see how that work pans out.
Yeah, so I don't have it from a comparative perspective, but I can tell you that I have some unpublished data, that manuscript's been sitting on my desk for about a year on red-tailed hawks, and their immune defenses change. Their immune defenses are different depending on whether they're coming through in an early part of the fall migration in California or the later part of the fall migration, and it does have effect on their survival probability, but it's hopefully going to a journal in the next couple of weeks. I just have to get it off the desk, but so there is some evidence, right? And I'm obviously not the only person to have asked about that. The red knot work is really great on migration and immune responses.
Alan Berkowitz has a question from Carrie's staff, so I'm gonna see if this works. Alan, are you there?
Am I here?
Yeah, you are.
Can you hear me? Or
yep, we can hear you.
Okay, so I guess I just so you showed all these relationships, and I kept waiting to see an R square, and because that's what you know, and it's so cool to be so focused on the slope and the kind of the range of slopes. But I guess I wondered, it seems like most of your R squares are probably pretty small, so there's kind of big other things going on.
There is.
And I'm wondering, especially, so in general, kind of, what are your avenues of kind of trying to pursue those? And I guess even the migratory versus non-migratory, that seems like an example of that. But then I also wonder the implications of that for making individual species comparisons, which it seems like could be pretty interesting, like the elephant versus the mouse or something like that. If they fall on a very different place on your relationship, right? Then your relationship is not necessarily explaining their difference that much anyway. You see what I'm trying to get at?
Yeah, I absolutely do. And thank you. We did calculate the equivalent of R squared for these models, and for the heterophils. Actually, hold on. I think I have. Oh, I forgot to thank all my partners. My partners have been really critical to this. Sorry. So here are the data from the neutrophils again for the mammals, and the breakdown of what explains the variation, and the evolutionary relationship in this for this immune response is overall explains the majority of the relationship. And so mass explains about 9% of the variation, which seems like a small amount, but it's interesting from the perspective, it's explaining about 23% of the variation that's left over after the evolutionary relationship has been accounted for. And we also did a model where we included sociality, really broadly classified as solitary, in pairs, or in groups, and trophic level as a measure of diet, and life history characteristics. So we had a measure of survival, and we had a measure of reproduction, reproductive capacity, and all of those other traits combined explained another 9% of the variation. And so body size alone is explaining what those other characteristics combined can explain in the data set. I didn't present these data in the talk. These are from those same mammalian species. These are lymphocytes, and they scaled close to isometrically. And a body mass explained a very small proportion of the variation. This is the platypus, in case anyone's interested what that outlier is. We see similar patterns when we look at scaling of heterophils, and similar explanatory power when we look at heterophils in birds. And then when we look at the functional complement assay that I presented in the latter part of the talk, phylogeny explains, depending on how you calculate its effect, it's explaining about 20% or less than 20% of the variation in the curve parameters. It depends on which curve parameter you're looking at and how you do the statistics. And so it's explaining a remarkably small proportion of the variation, and it was surprisingly small. And body mass is explaining, as I said, it's isometric. And so it's explaining a very small, it has a very small effect size, right? And we have not done an analysis looking at life history and ecological effects driving those relationships and explaining the residual variation. Based on the other scaling that we've done, I suspect that, and given how closely life history is tied to body mass in mammals, I suspect it might not be explaining a huge amount of the variation. We have started doing some preliminary diet looking at, or data or analyses looking at diet and things like whether they're carnivores or omnivores. So these really large diet classes, because before it was trophic level. And that does, there is differences in how body size, there's an interaction between body size and diet type. And that is an analysis that my collaborator is doing, and I haven't seen the final results, so I can't do anything but tease on that. But yeah, I do think that this data set heading forward provides an opportunity for collaboration and asking about other ecological traits and how they explain residual variation. Thanks for the question.
We have another question from Jane Lucas, one of the scientists here. Jane, can you hear us? Maybe not. Oh, there we go.
Hey, can you hear me?
Yep.
Okay. So maybe I don't quite know how you're completely classifying parasites, but one of the things I was wondering is if you've looked at the size of the microbiome in these different organisms and like kind of the microbiome load, and whether or not host immune systems are really good at telling the difference between what is kind of parasitic versus beneficial, and whether or not you've looked at all about kind of that trade-off from the microbiome perspective. Hopefully that makes sense.
Yes, it does. So it is something I'm really fascinated by, and I actually don't know how to do the microbiome analysis. So I'm looking for a collaborator if anyone's interested, but I'm actually sitting on data from a whole bunch of, either we've collected it, gleaned it out of the literature, or it's de novo collected with my colleague at Hamilton, or my former colleague at Hamilton, Mike McCormick, on microbiomes of zoo animals and ranging in body size. And I do have questions around things like, if there's a greater how do I want to phrase this? If there's a greater sort of absolute quantity of microbes in, for example, a digestive tract, do they perform and that scales at a relationship where it's disproportionately high in large organisms, and digestive tract length scales isometrically, but the surface area scales hypometrically, then that means there should be a thicker layer of microbes at each area of cell wall, and that might provide sort of passive protection. And then, as you were mentioning, functional groups. And so I have some data that I would love to start thinking about and exploring, but COVID really slowed down how we move things forward. And so that is something that we've been thinking about, and it's just been sort of tangential to the main parts of this project, and haven't had a chance to look at. But I think we're talking later today, Jane, and I would love to chat with you more about that.
Oh, that sounds great. Yeah, I think it would be a very interesting avenue, and I think people have not explored a lot in the world of, like, yes, microbiomes are beneficial, but they're also there is an associated cost to maintaining them, and you might have some really interesting data to address that.
Yeah, and I mean, my other line of research is really around trade-offs. And so I'm also thinking about, are there trade-offs as organisms are going from small to large, and are they mitigating those trade-offs differently, right? Are they making fundamentally different trade-offs? And that leads to questions around neutralisms versus commensalisms versus parasitisms, right? And I would love to chat with you more about it.
Awesome. Great.
I think we might run out of time, but we have a couple of questions in the Q&A box. There's one from Z. Grabowski, who's a postdoc at Carey Institute, and he says, he asks, curious if these data sets would be useful for examining behavioral aspects, such as those that would modulate exposure, including inter and intra-species grooming, or behaviors related to like self-medication, I think, to follow up on the diet discussion.
Yeah, that's a great question. So the short answer is yes. The long answer is I think that where we've integrated behaviors so far, we've really been focused in on the kind of physiological immune system in our data collection, but we did integrate data, our behavioral data, in our framework, which I should acknowledge Barbara was a co-author on that paper, in the Trends in Parasitology paper, where we developed a scaling framework for, or extended, because other people had started it, scaling frameworks for thinking about host competence. And behavior would really affect sort of that exposure and susceptibility and transmission stage. And so we did have things like grooming behavior and social behavior built into those models for our 3 example microbes that are pathogens we worked through. I think that those data, behavioral scaling data, there's some work, Candile published a really nice paper in Trends on it, but I think that behavioral data isn't as well quantified from a scaling perspective as sort of these count data, like number of eggs produced, and these physiological data that have been studied for 100 years. So I do think that there's a place for that, and I hope people are doing it. I think it would be great.
Behavioral work is really tough.
Yeah, comparative behavior work's hard.
Yeah, for sure. So maybe we can squeeze in this very last question that's from Rick Ostfeld, and he asks that, he says that there's some theory and evidence, which I know you're familiar with,
Yep.
Cynthia, suggesting that small-bodied mammals are restricted in investing in metabolically expensive aspects of immunity, but less so in inexpensive arms of immunity. The complement cascade might be less expensive than some induced aspects of immunity. Could that explain the isometric scaling of the complement cascade work?
You know, I hadn't thought of it from that perspective, which I would expect me to have done. But I actually think that you might be right? I've been thinking about constraints on the large, or what maybe. I haven't been thinking about constraints on the small side. I've been thinking about maybe why large organisms, maybe they're trading off. I think large organisms might be trading off some sort of, how do I want to say this? Right, they have a lot of neutrophils, but we don't know if each of those neutrophils is as functionally active as a neutrophil from a mouse. And so I've sort of been thinking of it because of the neutrophil data from that perspective. But you're right, it equally might be a trade-off or a constraint on the small body size, that maybe mouse cells are already sort of running at maximum, and elephant cells have the propensity to upregulate and downregulate depending on their exposure. And that's an interesting idea. I think we're talking later. I would love to talk to you more about that too.
Yeah, thanks so much for all of this. Really, I mean, obviously folks have lots of questions, and if you're around the building today, Dr. Downs will be joining us this afternoon from 12:30 to 2. She'll be available to hang out and answer questions. Thank you so much, Dr. Downs, for such a great seminar, and thanks everybody for attending. See you soon.
Thank you so much for having me.